From this class onwards, now we will move on to 2D NMR spectroscopy. Till the last few part of the course we have looked at 1D NMR. So, now let us start with the important aspect of NMR spectroscopy, which is 2-dimensional NMR. Here we are going to see how a 2D NMR spectrum is recorded, what are the basic principles involved in 2D NMR and how it can be interpreted, and also in the last part of the course we will be looking at how we can use 2D NMR for different applications. Typically 2D NMR applications is basically in the case of organic molecules very useful, and also it is very useful in the case of peptides which are made up of amino acids. So, we will see that in the last part. Right, we will now get start with the 2D NMR basics. So, you can see, as we saw here in the case of 1D NMR, there are few limitations of 1D NMR which you cannot, some information which you cannot obtain from 1D NMR. So, let us see what are those drawbacks of 1D NMR. Number 1, it is not possible to correlate two different chemical shifts by 1D NMR. So, what do we mean by this? What it means is, for this you have to imagine the 1D spectrum, let us say of a ethanol molecule where we have a CH2 CH3 for hydrogen. So, now in case of ethanol is very straightforward that there is a CH2 system H2 and there is a CH3 H3 and they are J coupled to each other, and one gives you a triplet, another gave us a quartet, right. So, CH2 gave us a quartet and CH3 gave us a triplet. So, that was simple. But now if I go to some other molecule, we will see down the line some examples. But if I go to some other molecules where there are more than these pairs of protons, so CH2 CH3 I call them as a pair. So, if I see such multiple pairs in a proton spectrum, I do not know which pairs are correlated with each other. So, which CH2 and which CH3 are correlated with each other. So, that information is not obtainable from 1D spectrum. So, one has to therefore use a 2D NMR which will help us to know which pairs are possible. So, let me illustrate this with a picture we can look at a schematic drawing. For example, let us say I have a hypothetical molecule where I have a CH3 and a CH2 somewhere in the molecule, and somewhere down the molecule. So, this is the same molecule, I have another CH2 and another CH3. Okay, so, there is some hypothetical molecule like this. Now, what will happen is this CH2, this proton is coupled to this proton. So, you expect a triplet for this, a quartet for this, but at the same time similar thing is happening here. Here also I get a quartet for this proton and I will get a triplet for this proton. So, if I record a spectrum, I will get two triplets in the methyl region and I will get two quartet in the methylene region. So, now I do not have information that which two are connected to each other, I do not have that information. So, that information of connection between which methylene is connected to which of its respective methyl, that information is not present in the simple 1D NMR. All the 1D NMR gives me is simply a set of lines. It tells me there is this is a methylene group, this is a methylene group and so on. It does, for example, it tells me that this is a methylene molecule, methylene moiety, this is a methylene moiety, this is a methyl and this is a methyl, but it does not give me the information of the connectivity, that means which methyl is connected to which methylene. That information connected in the sense like this connection coupling, that information is not provided in this 1D spectrum. So, if you want to know which methyl peak here is matching with which methylene peak here, which are the coupling partners, you will not get that information, because this will also be a quartet, this will also be a quartet, this will be a triplet, this will be a triplet because of the J coupling, but that connection is not there information. So, that is for that we need to go to 2D NMR. So, this is one of the limitations of 1D proton NMR. And secondly, you can resolve overlaps. So, again we will see some examples how we can resolve overlaps. So, what happens is when you have a large number of peaks in a proton spectrum, the peaks will start overlapping, they will start crossing each other, they will lie on each other. So, there is a lot of degeneracy. Degeneracy is the word used for saying that there is an overlap of peaks. So, to resolve those degeneracies or to resolve those overlaps, we need to go to 2D NMR. And a third aspect is more technical point. The point is that in NMR, which we did not see because of that will be not possible in this course to deal with, we will look at that in an advanced version of the course later. The thing is that you have what is called coherences in NMR. Whenever you apply NMR pulses, you generate coherences. So, if you have two protons coupled to each other by J coupling, there is a possibility of generating multiple quantum coherence. So, I will not go into detail of this terminology because this is not part of this course, but as information is useful to know that multiple quantum coherences cannot be detected by NMR. They can be generated, but they cannot be detected directly. So, to detect multiple quantum coherences, we have to do it by 2D NMR only. In 2D NMR, what we do is we generate some multiple quantum coherences, and when we want to detect them, we convert it into into a single quantum coherence. So, for the time being, you if it is okay if you do not fully understand these concepts of the multiple quantum coherences, but remember in mind, keep in mind that 2D, one of the reasons 2D NMR is very useful is that you can indirectly detect multiple quantum coherences, because in a 1D you cannot directly detect them. So, now let us see and look at these three of this, at least two of these two cases, how and what 2D NMR helps to resolve. So, let us take this example again, this molecule which we saw in the previous class when we were looking at carbon 1D NMR analysis. There we saw that we had a 2D spectrum, a 1D spectrum of hydrogen, and we also had a 1D spectrum of carbon. But the one question which I raised last time was that there is no connection information in from these two. What is happening is we have recorded these two spectra separately in isolation, means this was recorded initially, then you record this, but I do not know which proton peak here corresponds to which carbon peak here, because all of them are CH peaks. But of course, you can say by logic, again if you use a logical idea, you can say that look, this will correspond to this and this will correspond to that and so on, but that is based on some kind of a logical arguments. But if I want to directly see that which proton peak here, this is let us say one proton peak, because this together is 1 and this is a doublet, this is 1 and this is another 1, this is a bunch of 1 peak corresponding to a J multiplet. So, this particular peak, 1 peak corresponds to which carbon here. Of course, remember this is coming from ketone. So, we are not going to worry about the ketone because there is no proton attached for ketone. So, I am not looking at the ketone, I am only looking at these carbons, and again this quaternary carbon, remember, is not, sorry, this is a proton here, single proton. So, there is one carbon, these two are equivalent. So, 2 carbons and this is a third carbon. So, we have 3 carbons here. Similarly, there are 3 protons here, because there are these methyls, these 2 methyls correspond to 1 methyl, this corresponds to this methine is 1 hydrogen, 1 type, and this is a third type of hydrogen. So, 3 hydrogens. So, we have 3 hydrogens on this side, 3 carbons on this side. So, can we match from 1D which is which? That is a question which is shown in this diagram. For example, can I say that this particular doublet, 1 hydrogen corresponds to which carbon here? That information is not present in this 1D, and this is one of the drawbacks of doing at 1D separately. But if I do a 2D, I should be able to find out. So, that is the question. So, suppose I label this, let us see how a 2D will help us in this information. So, right now again keep in mind we are only going through idea of how 2D can be, how 2D can help us to resolve. As we go along, we will see more details of 2D. So, in the next slide we will see a simple 2D, but I am not going to right now, we will not see how the 2D is recorded, we will come to that later. So, let us now look at how a 2D can help for this problem. So, what is the problem in hand? The problem in hand is as follows. We have 3 hydrogens which is labeled ABC, and we have 3 carbons which is labeled XYZ. Now, my job is to match, or my problem is to match which proton here is paired with which carbon here. So, there are 3 pairs possible. means 3 pairs are there, which pair is the correct pair. So, for that is what is shown here. If I try all possible combinations, I have 12 combinations possible, because this can go with any of these 3, this can go with any of these 3, this can go with any of these 3. So, there is basically there are 9 possible, but there are actually 4 peaks in the satin here. So, if you see there are 4. So, of course, as I said, by logically you can say that this is not attached to a proton. So, you can ignore that combination, but if you take brute force, a mathematical approach tells me that given 4 peaks here and 3 peaks here, I expect 12 combinations or 12 pairs, okay. But we do not know which pair is correct, but only 3 pairs are correct, because this has to be paired with one, this has to be paired with another carbon, this has to be paired with another. So, only 3 pairs are the actual pairs, but based on this spectrum separate alone I am not getting that information. So, that is what can be seen. Suppose I do now a 2D NMR. Okay. So, if I do a 2D spectrum, that means on this axis I have put the proton and in this axis I have the carbon. Okay. Now, this is the 3 pairs. Okay. So, this is already known to us, but I am just trying to show here that if I do a 2D spectrum, I will get the correct spot, because the correct peak which is in the carbon side here, which was coming at 20 in our previous slide, will show up now its correct partner from the proton side. So, you see this is simple 2D coordinates, this C comma Z is nothing but is telling me that the proton number C, which we saw in the last slide, which we assigned as C, that has this partner in the carbon side corresponding to Z, which was in this side. So, a 2D spectrum, if I record, immediately out of those 12 combinations gives me the right combination, because it connects the right carbon with the right proton. How it does it? We will see that later, but the advantage here of 2D is that out of the 12 combination which we saw, I am able to get the right combination simply by recording a 2D spectrum, and the right partners, the combinations are showing up here. So, similarly, if you look at the second combination B to Y, it comes in the 2D spectrum, because that is the only one which matches with this peak and corresponding to its partner. If you draw a line and a straight line here, that is that peak which was a 2 ppm value, and that now corresponds to this carbon. And the last combination was the septet. So, if you recall in the last slide, we saw a septet, and the septet was basically at around 3 ppm, 2.5 to 3, and that septet is what is coming here, and its corresponding carbon is basically here at 40 ppm. That is what is shown here. So, this is basically what we saw, that if you see x, y, z, the A matches with the C matches with Z, okay, and B matches with Y, and A matches with X. So, this is basically what is how the 2D information helps us to resolve the ambiguity. So, what was the ambiguity? The ambiguity from the 2 separate 1D was that we do not know which proton peak matches with which carbon, which 2 carbon proton are paired with each other, but from the 2D that pairing directly comes, because that is how the 2D is done, where the correct pairing shows up by a simple this axis. So, on the y axis is your chemical shifts of carbon, and x axis is the chemical shifts of hydrogens. So, this is what we say correlation. So, the word correlation is used in NMR, 2D NMR very often. The idea here is that we are correlating the 2 frequencies, means we are connecting or matching the 2 chemical shifts. One is carbon chemical shift, is hydrogen, and by connecting those 2, I am able to figure out which proton is correlated with which carbon chemical shift. Okay. So, this is an example of what is called as a heteronuclear NMR experiment. Why hetero? Because we are connecting a carbon with proton. If I have proton on both sides y and x, we will use the word homonuclear. Homonuclear means same nucleus, same type, that is hydrogen and hydrogen. If I use heteronuclear, means it is hydrogen and carbon. It can be carbon carbon also. So, carbon carbon on both axes can also mean is a homonuclear. So, remember again, I have not generated this 2D spectrum from my 1D spectra. 1D spectra was done separately, A 2D spectrum is recorded separately. So, please do not confuse that this 2D spectrum which I gave an example is coming from the combination of 1D which we saw in the previous slide. That is a separate thing, we are recorded separately 1D, but when we record a 2D separately, this is not generated by combining the two 1Ds, it is generated by a different method which we will see. By doing a 2D I am able to get that ambiguity resolved. So, let us see at the second. Okay, so, this is what is shown here, that is what I said, that you are actually correlating two different types of nuclei. So, we use the word heteronuclei. Now, let us look at again this example in a different way. So, there is one more case. The question is which protons are coupled to which proton. So, this is a separate problem. In the first case we saw which proton is coupled to which carbon, that is what we saw in the previous slide. For the same molecule now, now we will not look at the carbon, we are looking at only the hydrogens. Now, can we say that A is coupled to B, because of this it is getting this multiplied, or is it that A is coupled to C? Now, which is A, which is B, which is C, we do not know, we just label these 3 protons, 3 peaks as A, B and C. So, the question we are trying to ask here is that given a peak doublet here, means it is coupled to some proton, it cannot generate a doublet if this is not coupled to anything. This for example is not coupled to any proton. So, therefore, there is no coupling of B to any of the protons, but C should be coupled to A, only because of that it should be able to get a doublet. Similarly, A should be coupled to C, only then it can give a multiplet. So, this information of which two hydrogens, these are all hydrogens now, which two hydrogens are coupled to each other, that information is not coming from 1D. In this case, yes, in this case it is easy to say, because this B is not coupled to anybody, therefore, it is a singlet. So, I can say conclude that yes, A should be coupled to C, but remember in a more complicated system this information is not going to be there, and therefore, it is required to do a 2D NMR to give you this information. So, let us see how 2D NMR gives that information, which is shown here. So, you see if I record a proton to proton spectrum, means both axis is proton now or hydrogen now here, compared to the last example where we saw carbon was shown here, but now we are looking at hydrogens on both axes. So, this is a 2D homonuclear NMR experiment, and that type of experiment what it is doing, it is correlating, again remember the word correlation, it is correlating a proton with another proton, and how these protons correlated, it is taking a proton which are coupled to each other are correlated. So, if you see here this proton which is at 1 ppm, which is a C, it is showing that that is correlated to the proton which is A and the x axis. What is A? A is this proton. So, that is what we expected based on our logical arguments, that B is not coupled to anybody, because it is an isolated CH3 is shown here, but A and C we expect to be coupled to each other, of course. So, that is why it was easy to see from 1D, but the example here is to show that a 2D gives you that information. So, in this particular case the information was obtained not directly, but by our logical arguments, but here the information is directly obtained, and you can actually see in this particular case that you can see that there is a coupling between, there is a correlation between the two protons. So, for example, if I have many CA pairs, okay, so in this case there was only one A, one C, but remember I showed you in the beginning of the class today that you can have many CH3 CH2 groups in a big molecule. So, you will have many such AC pairs, you may have many isopropyl groups, right. You may not have only just one isopropyl in a molecule, you may have multiple isopropyls. So, in all those isopropyl groups, how do you know the CA pairs? Because you will get many of these doublets, you will get many of these septets. So, which septet is coupled to which or connected to which doublet, that information you will not get from a 1D, but that you will immediately get from a 2D, because the septet which comes at this position will be automatically or correlated with the doublet at this position. So, if you have many doublets here and many septets here, they will get individual peaks. This is called a peak. So, you will get individual spots here for each septet doublet combination. So, that is how 2D NMR is another example how it helps us to find out which protons are coupled to which protons. So, this is the first point which we saw, advantages of 2D. Now, let us see the next example of 2D which helps to resolve overlap. So, this is what I was saying in the previous, that B is not coupled to any hydrogen, therefore, it is absent in the spectrum. So, this type of experiment is called homonuclear. So, two homonuclei are coupled to each other or are detected on both axes. So, it is a example of a homonuclear experiment. So, now as I said, in a simple molecule like ethanol, it is very simple to say that okay, these are the two coupled to each other, because there is only two hydrogens seen in the spectrum. But look on this side, this is a complicated molecule, it is a protein molecule which is made up of amino acids, and all the hydrogens which are shown here by grey color balls, all of them will get a peak here. So, you see it is a very complicated spectrum, it is not very simple like this. So, therefore, for such complicated spectra or spectrum, we cannot use simply say which hydrogen is coupled to which hydrogen. In fact, we cannot even see the hydrogen peaks properly. So, to resolve overlaps, to get that correlation information, we have to do 2D NMR. There is no choice for us for a bigger molecule like a protein or a peptide. Even for a small organic molecule like a drug molecule, which may be the molecular weight will be around 500 or so, even for those kind of molecules, we need to do 2D homonuclear or heteronuclear spectra, so that we can get the information of which coupled peaks are there here. Okay. So, let us look at another example how we can see resolve overlap. So, this is a very interesting example which I am going to talk about. This is just a hand waving argument, this is just example from not from NMR, this is a non NMR approach. So, let us say that you have a name of a person called Anil Kumar. Okay. So, this is actually a name of one of my colleagues. So, I am just going to take that example. So, let us say I am trying to search the phone number of Anil Kumar from a directory. So, let us say I look at only the surnames. So, if I look at surnames, obviously there will be number of Anil Kumars, right, number of Kumars. There need not be Anil Kumar, there can be many other Kumars. So, you see this is what I have depicted schematically here, saying that there are many Kumars and they are all having in the line, because that is called overlap, they are all overlapped. So, I do not know obviously which Anil Kumar, there are many Anil Kumars. So, I do not know which is many Kumars. So, I cannot simply look at the Kumar and get the phone number. Let us say I look at the first name, suppose I search in the directory by another approach, I start looking at the first name, again there will be many Anils in the directory. So, I will not be able to figure out which Anil is the one I am looking for. But if I combine the two, so this is what is shown here, many first names are Kumar, many surnames have, sorry, many first names are Anil. So, let me correct it, here let us say Anil, okay, and many first surnames are this one surnames Kumar. So, they overlap. Okay. But now, if I combine the two, it is very obvious that there will be probably only one Anil Kumar, or there may be many Anil Kumars, but at least now by combining the two, correlating the name with the surname. So, you see now the correlation is coming out of this example. So, what I am trying to do, I am correlating or connecting the first names of people with their surnames. So, when I do that correlation, I get a unique combination, because there may be only one Anil Kumar in this directory, or there will be, even if there are several, at least I have separated them out of the unwanted names which are having different first name, but they may have surname as Kumar, or they may have first name as Anil, but different surnames and so on. So, by using this kind of a correlation idea, I have now reduced my ambiguity, means I have reduced my problem to a very simple problem, that now I am focusing only on the combination. So, this is basically what is the idea of 2D NMR, that it helps you to resolve that you can, let us say you resolve the peaks. Let us say I connect the chemical shifts. So, in the earlier examples we saw, we use chemical shift in both the axes, we use carbon proton on one side, carbon on the xy axis. So, you see by combining the carbon and proton chemical shifts, I was able to get a unique combination and I was able to resolve the overlaps problem by going from, instead of I have 12 combinations which were possible. If you remember, we saw that in case of 3 protons and 4 carbons, we had 12 combinations, but instead of 12 combination I reduce the problem by going only to 2 combinations. So, by 3 combinations. So, this is how was an example of how we can resolve overlap. So, similar is an example shown here that NMR 2D NMR helps us by correlating the chemical shifts from 2 dimensions using 2 dimensions. So, this is basically how 2D NMR spectrum is recorded. So, how do we actually practically do this experiment? So, now we will look at the details of how a 2D NMR is done in reality. So, we will go through several different ways of explaining how 2D is done. So, I would suggest that you have a look at all the possible explanations. So, we will see the first one how it can be done. So, the first thing what we do is that we start with an RF pulse. So, this is similar to what we do in 1D NMR. We apply an RF pulse, okay, that is called excitation. So, we excite a nucleus from alpha state to beta state by applying an RF pulse. So, that is the first thing which we do in any NMR experiment. So, same thing starts here also in 2D we start with that. Then what we do is at the moment you excite a nucleus, then you leave the RF pulse, you remove this RF pulse, then the nucleus starts relaxing, and that is called FID. During that period, remember we said in 1D you generate an FID, you induce a signal in the coil and that generates an FID. But in 2D we do not do that. In 2D what we do is we allow it to evolve. This is the word used, evolution. Remember, please do not confuse this with the evolution we use in biology, that is called Darwin's evolution. That is a completely different concept. This is just the word is somehow used for evolving a chemical shift. That means is that the FID, the chemical shift oscillation will start after you remove the pulse, and this is what is called, this is because of the relaxation and chemical shift. Now, during this process when it is going relaxing, during that process, that means from here to here, this is, remember, a time axis. Okay. So, this is x axis, here is a time. So, during that process what it does this nucleus 1 transfers its magnetization to another nucleus while this is happening. Okay. So, that is what you have to keep in mind. We will again, as I said, we will go through this exercise of understanding 2D in different ways. So, this is one way to understand, is that we have a nucleus which is excited, it is now evolving or moving in chemical shift because of which you get these oscillations, and during that process it is also relaxing, that is why it is showing a decrease. During this process when it is moving in chemical shift as well as relaxing, it transfers the energy to another, or transfers the magnetization to another nucleus. And why is that so? Because it is coupled to this nucleus. So, let us say these are 2 hydrogens coupled by J coupling or some other coupling to each other. Because of that coupling they interact with each other, and because of that interaction they transfer energy to each other. So, nucleus 1 will give a magnetization to 2, 2 will give it to 1. So, we are only looking, let us say, one side. So, the nucleus 1 gives its magnetization to 2 during this process. So, remember we are not doing any extra work here, all we are doing is allowing the nucleus 1 to simply move in chemical shift and relaxation. During that process it starts transferring the polarization to B. Now, that transferred polarization is now detected by exciting this nucleus. Remember, this nucleus it has only transferred, but we have not excited this nucleus. So, we have to excite this nucleus. When we do that excitation, that nucleus now will start moving in its chemical shift, but it will carry the information of the chemical shifts coming from here. So, these two informations are coupled to each other, and what is done is, this is what is detected physically by using a receiver. So, this is like the 1D NMR where we detected the signal, but here we did not detect, we simply allowed it to evolve, we say, with chemical shift. During that process it transferred, we then excited this nucleus and detected its signal, but that signal what comes out is having the information of this. So, finally what we do is we do a Fourier transform of this two spectra. Remember, this is not two different spectra, they are within the same experiment. So, this is you can call it as one frequency dimension or axis, and you can say this is another frequency because another chemical shift is there, and that is frequency 2. So, by doing this Fourier transform, what we are going to get is that we are going to get this information of this chemical shift directly connected to this chemical shift, that is this frequency, and that connection is not possible if I do a separate 1D here and I separate 1D here. But because of this connection, because I am directly going from one nucleus to other, I have actually got them connected to each other by J coupling, and that connection shows up in the 2D spectrum which is shown here. So, with this idea we will again go through this exercise in the next class, and we will continue with this 2D NMR, and we will see how further we can understand how 2D NMR experiments are recorded and how this frequencies 2D axis can be interpreted.